like the stem, or like the leaf, or like the root. Okay. The only thing is that we are adopting different means to do it. All right, now, like you have already examples of grafting, you have examples of cutting. Like in the case of rose plant, we simply cut the stem of the plant and then plant in the soil. Sometimes it is first kept in a solution that is having rooting hormones. So thereby hormones, the rooting hormones will help to develop the roots in the system, and then we plant it. Then you have got the layering system also, right? So where we get to see, like layering is done for the runners, the plants which are runners like this, like the example of a strawberry, chrysanthemum flower, peppermint, or the grass. So those are the runners. uh, for the food crops, that for the food crops, basically artificial layering will be done, right? Now, or for the ornamental flowers also, the artificial vegetative propagation is done. Because for the wild crops, like wild plants like grasses, there's no need to there's no need to artificially grow it, right? Now it grows on its own,
Yes.
right? Then you have got the tissue culture also, right? So all these things we are going to study starting from the grafting. Many times, like definitions is also asked in the exam, like what is simply artificial vegetative propagation. So we will answer it like this, like the we will answer it like this: the type of vegetative reproduction which is carried out by the humans in the fields and laboratories. Okay, so that will be artificial vegetative propagation. The most common of them are as follows. You have the example of cutting. So cutting, who would like to explain it first? Any who has actually any idea about cutting, like how it is done?
So like for example, rose.
Hmm. For example, rose, good example. So there, what we do, we simply cut a vertical section of the stem, right? And then you plant in the simply soil. But then we need to ensure few things. Like see this diagram, you can see there are nodes growing on the leaves, right? This
Yes,
structure
sir.
that you see, these are the nodes. Okay. So nodes, we need to show that there is a at least one or two nodes still attached to the stem. So then you will simply cut the stem from here, from any part, and then you will plant it under the soil. Sometimes we first keep it in a solution that is having some rooting hormone in it. That is having rooting hormone in it. So like naturally also the roots will eventually grow out from that cut portion of the plant, from this cut stem. But if you were to first dip it in the rooting hormone, no, and then left it for a few hours, so thereby the roots will develop more quickly. Yeah. So that is the cutting over here. So when we plant any part of the plant, like a stem, like especially the stem, by cutting it, then it is called the harvesting of that crop in that manner. Sometimes cutting are also used for root development, like this one which I just told you. And this cutting is a asexual method, right? It's a sexual method of reproduction, right? So therefore, since it's a asexual method, no, the new plant is being developed from the same plant. So thereby, genetically, both the plants are identical to each other, right? So in common language, it is called as clone. Here, the second part, that is basically cut from the original parent plant. Both of them are cloned to each other. So we do it for plants like roses, right? We do it for bougainvillea also. It is done for this plant also. Let me rewrite that. Anyone like you guys know of some examples? Yeah, anyone who would like to give some examples for which cutting is done?
So, like money plant.
For money plant, huh? For money plant also it is done. Good. Yeah, anyone is having any questions here?
No, sir.
No, sir.
Also, one important question here is that you guys tell me, like this new plant that has developed now, has it actually developed from radical, huh? Or has it actually developed from the adventitious roots?
Sir, I didn't get it.
The question here is that the new plant, this new plant that I am growing here, the roots of this plant, will it be has it developed from the radical, or has it developed from the or is it a type of adventitious roots?
It's a radical.
Sir, it's radical.
No, listen to the question again. When I will be cutting a portion of this of this plant, and then I will be planting the stem into a pot, so there will be obviously development of roots, right? So do tell
Yes,
me,
sir.
this root, did it develop from the radical, or is it adventitious roots?
So it's adventitious roots.
Adventitious roots. Now adventitious roots are what? The roots that develops from parts of the plant other than the radical.
Yes, sir.
Again you guys tell me, what was radical and plumule which we studied? Yeah, when a seed germinates, there are two different sets of tissues that comes out of it, right?
Yes, sir. Like one is
Yeah.
radical and one is plumule.
Hmm. Which one of the two is radical, which one is plumule?
So the one, the second one is radical, right, sir?
Exactly.
And the first
Exa-
one is a plumule.
exactly. So radical later on develops into root actually. Yeah. So for the original plant, na, I might have grown it from the seed. That seed germinated, so thereby root originated from the radical. But for the clone plant here, the new plant, it actually develops adventitious roots, right? Now? Okay, so this is all about cutting. Has anyone any doubt in it? Please do let me know.
No, sir.
No, sir.
No, sir.
Then we come to the second method of artificial
Sir,
vegetative propagation. Yeah.
we have explained this grafting. We have explained it.
We have discussed that. No, I don't
Yes,
think
No, sir.
we have discussed it.
we just like in just
Uh,
brief we discussed it. I think in brief we discussed it, right? But not in detail. Okay, now. So here we'll be discussing it in detail, even if I touched this point, but I will be discussing in detail here. Okay, now. So look over here.
Yes, sir.
Grafting is a method in which two plants are combined, as you can see, to form a new variety. Like this plant could be, let's say these are two plants. Say these are two parts from the mango plant. Both of them are, say, this stalk. It belongs to a mango plant. Okay, whose mangoes are bigger in size. All right. This plant, this part, um, say this stalk, I have taken it from plant A, right? And plant A actually gives bigger size mango. See the second part over here, that is scion. Okay, I have taken it from plant B. Let's say that it gives sweet mango. It grows sweet mangoes, all right? Okay, so I have taken the stalk from a plant whose mango size are very bigger. The second part of the second part, that is scion, I have taken it from plant B that bears sweet mangoes. Also, this is actually that part of the plant that is having the root system. Okay, now, while the above portion is not having the roots in it, you have just cut a stem from the plant. You have taken a vertical stem from the plant. While the stalk that you see, you are having the plant in which roots are still attached, right? So now I have joined them together. Right now I have combined the two mango plants having different varieties of mangoes growing it in it. So now the mangoes that will grow on this plant, it will be of new variety. So what kind of new variety? The mango that will be grown on this plant now, it will be big in size plus sweet also, right? If
Yes,
earlier,
sir.
if earlier the mangoes that were growing on this was small in size, right? And now earlier, when I had the plant A, the mangoes growing on it were uh not sweet, lesser sweet, right? So now when I joined the two plants now, I saw that their properties are combined together. Like the mangoes that will now grow on the plant will be bigger in size plus sweeter also. Are you guys getting it or not?
Yes, sir. We got it.
Sir,
Right?
get it.
Look here. It will be clear. Look here. What we have done here now, Chakrika, here's a plant. Okay. See, this is a plant over here. Say there's another plant here. Yeah, Sundus, by the way, is it clear to you?
Yes, sir.
It's plant A, it's plant B. See that the mangoes now that were growing on this plant were bigger in size. They were this much bigger, right? While on this one, they were only, say, this small only, right now. But they were small mangoes, but they were sweet in taste, right now. So one thing is good, but the another thing is bad, that they are very small in size. While the mangoes that were growing on plant A, now, see, they are very bigger in size, but sour in taste. Let's say, yeah. Okay, Chakrika.
Yes, sir.
Now, what if I were now to take the root system of this plant, the one that is having, that is bearing mangoes of bigger size? I will now remove this much part. Okay, I will be removing this much part of this plant here. No, we will be doing it on this one. No, look over here. I will have to draw it again. See, this one is bearing mangoes of bigger size. All right, no.
Yes, sir.
Okay. No, that was actually correct. Huh. So I will be now taking the shoot system of this plant. I will be cutting that out, and now I will take the shoot system of this plant, and now I will graft it over this plant A. I will graft it over A. Now remember the properties of the plants. What were the properties of the plants?
They should be of the same like
Hmm.
plant.
We should.
Like they would be if the one is mango, then like if the one is mango, then
Mhm.
other would be also
Exactly. Exactly.
mango.
Another would also has to be a mango plant. You can't be doing the grafting in a mango and in a lychee plant. Both have to be the same plant, right now. Okay. Now, Chakrika, you see here, all of you see here, the plant A were bearing mangoes of bigger size, while the plant B were bearing mangoes of sweet. They were bearing sweet mangoes. So now when I have grafted them together now, so then what will happen? It will be growing bigger mangoes plus sweeter mango also. right now, because the properties of the two have integrated here. They have combined together to form a new variety of mangoes that are bigger in size and sweeter as well. Hope it is clear.
So, but like the bee
Mhm.
plant, it should be like for that one, the mangoes, if they grow again, they'll be sour, right?
No, the property of this here, the mangoes that are growing, they are sweet now. Yeah,
So,
they
like
are having
the
sweet
top
now.
part of the A plant will
Mhm.
graft it to the B plant, like the
Hmm.
branch of the B plant.
Exactly.
So
So
then what about the B
Mhm.
plant, like the upper part?
we have taken the upper part of the bee plant over here. We have not taken the root system. We have taken the shoot system of the bee plant.
Yes,
For
so
bee plant,
like
you
what will we do with that then?
look here. No, we will be taking the shoot system of the plant B. We will be taking the root system of the plant A. Yeah, the variety of the mangoes that are growing on them are different now. So now when I will be grafting plant B on plant A, thereby a new variety of mango will be grown over this. So that will be bigger plus sweet as well. So both of their properties have actually integrated. They have combined here. So this is what basically happens. I have just taken
Okay, sir.
an example over here. For different plants, uh, different uh, different properties has to be considered, right? Like here we considered the
Okay, sir.
size and the sweetness or sourness of the mango here. So
Yeah,
here
so can
we
I have two
are.
minutes?
Yeah.
Can I have 2 minutes?
Okay.
Okay, sir. Thank you, sir.
Mhm. So in grafting, we saw that two plants are combined to form a plant having new variety. So in this method, any two varieties of the same crop, we see that they are being combined to form a new variety. And in this method, na guys, like any part of one variety of a crop, such as a stem, it is cut now and added to another variety of the same crop in such a way, guys, here that nervous system, like basically xylem and phloem, of both of them are being connected to form a new variety. One important aspect here now, like just like we have got veins, yeah, we have got nerves and arteries now. We have got veins and arteries now.
Yes, sir.
In the same manner, transport of food, transport of water, mineral and salts takes place via two vascular tubes. In the case of plant, they are mainly na xylem and phloem. Okay, so when you will having a vertical section of a plant like this or horizontal section of a plant like this, when you will take a vertical section out of it, you will observe that bundles of tubes are actually there. Not not literally like you will be cutting a plant, you will be able to see it, not in that manner. Okay, so I see that there are xylems and they are actually phloems. Right now, xylem and phloem. Xylem will be carrying water, phloem will be carrying food. Right now, as I said that here, what is happening? The two plants are having different kind of xylems and phloems. Now, this part of the plant that is cyan will be having a different type of xylem phloem. This one will be having a different type of xylem and phloem. Yeah. And now when I have connected them, now basically their nervous system has joined together to form a new variety. Is this thing clear
Okay, sir.
to
Yes,
this?
sir. It is clear.
Clear?
Yes, sir.
So this is basically what is happening here. What are some of the examples, by the way, in which
So, like
grafting is done?
for crafting,
For grafting, you
so
have an
like
example of a.
mango,
Mhm.
lemon,
For lemon also it is done. For like guava, right?
yes, and I guess some of the rose plants, etc.
Exactly. You can see like two different roses of two different colors are growing on the same plant. So
Yes,
that
sir.
is basically possible due to the grafting, right? So this
The apple also.
for apple also good.
Sir,
For apples also
Bongavilla.
it is bougainvillea, exactly, right? So
Sir,
for
Chico.
this, for chiku,
Yes, sir.
not entirely, not so sure about that. We'll have to check that. Okay.
Okay,
So
sir.
this much example is sufficient. Mango, guava, lemon, hibiscus, roses, apples, or bougainvillea. These are some of the examples of crafting. So while you were away, Chakrika, I just told one more thing. Like the two different plants here of the same variety, of the same kind, will be having different types of vascular bundles in them, right? Just like we have got nerves and veins, plants also have got xylem and phloem that transports food and water in their body.
Water.
So scion and stalk, they will be having different kind of xylem and phloem. But when I have joined them together, when I have done grafting, they will be combining together, right? Now, hence, due to that also, you will get to see like the um, fruits are of new variety here, right?
So basically, here in grafting, only the like the stock is changing, right?
No, the stock
I mean,
remains
the
same. Scion is changed.
scion is changing, right?
Exactly.
Okay, sir.
Okay. Then we study about the layering, right? So look, when you are doing the artificial vegetative propagation, like we were talking about the runners, yeah. Remember about the strawberry grasses.
Yes, sir.
So do we have to manually? Was there any manual intervention there in the case of the strawberry or in the case of like grasses? Were we manually twisting the branches or stems of the plant so that to allow them to touch the ground? Were we manually twisting the branches or stems to make them touch the ground in the case of grasses? Do we manually do that? No, obviously no. But in the process of layering now, what we will do, we will manually twist the branches or stems of the plant so that they will be touching the ground. So see the structures over here. What are these metal ring structure over here? They are there to hold the branches close to the ground. Getting it now? Yeah. Sundus, Vaiga, Chakrika, are you guys getting it or not?
Yes, sir.
It's a simple thing.
Yes, sir.
Hmm. So part of this plant that will
No,
be
but like, what if the stem breaks apart?
usually they are no elastic, a little bit elastic. Okay. It's not like they do not break. Sometimes they do break, but we usually do layering for the plants whose stems are elastic. They can be bent. You won't be doing it for mango or guava plant because they will basically break, and it in fact does not. In fact, they are not runners now,
Okay, sir.
right? Now we will be doing it for certain kind of plants only, right? Huh. Back to what we were discussing. So we get to see this part of the plant, the part of the branches that comes into the ground now. Now it is covered with soil. It has been now covered with soil. So thereby roots will start to grow from this part over here, right? And this root will be called as what? What do we call this kind of root? Yeah. What do we call this root here? Yes.
So advantageous roots.
Adventitious roots, exactly. Because they have not grown from the radicle, right? So they are the adventitious roots. You're getting it now.
Okay.
So basically, this is adventitious roots here. Okay. So here you get to see like roots will be extending from or growing from this structure from this part of the branch, other than the plant roots, other than the main plant roots, right? So this type of layering here, basically, the only human intervention here is that you have forcefully twisted the branch close to the ground. Otherwise, it grows naturally. Getting it now?
Yes, sir.
Now, in some cases, now you have like air layering also. Sometimes you will hear of air layering also. So air layering is what? Air layering involves now. It involves scraping and covering branches with plastic. You'll be covering the branches with the plastics in order to prevent the moisture loss. So due to and then what we'll do? Branches will be broken off and then replanted. What we will do? We will cut it from here. We will be cutting it from here, right? So that it now grows into a new individual plant. Yeah, you guys are getting it or not? Yes.
Yes.
Sir, I don't get it. Can you explain again?
Look, sometimes what we do now, basically, the branches are covered with polythenes. I hope I had the diagram here. Look, it is like this. We do air layering. So you might have actually seen like you in the branches now. We will do what? We will basically take some soil, like this, and then wrap this part of the branch with polythene, right? So
Okay,
after
sir.
some times, what will happen? Adventitious roots will start growing from this part of the branch. Adventitious roots start to grow from this part of the plant. So here we are doing the layering in air. In the process of layering, we covered a particular part of the branch with soil now, right? And then roots start to develop from there. So here we are doing that in ground now, right? Sundus, Vaiga, Chakrika. While
Yes,
when
sir.
you do this in the open branches that are in the air, basically, right? That is basically in the upper parts of the plant. So there I have taken some amount of soil. I have wrapped it with the polythene so that moisture is not lost and soil stays intact. So then after sometimes, after a few weeks, I will see that adventitious roots will grow from this part. So later on, I can cut it from here and then replant this part. So it will grow into a new plant. Yeah, is that clear?
Yes, sir.
Yes, sir. It's clear.
So I didn't understand.
Okay, Vaiga, please explain this.
Sir,
What
like
you
layering.
understood from it? This air layering, particularly air layering. This one. Yeah. Vaiga, you got this now. Hello?
Yes, I got like little bit, but I don't know how to explain it.
Okay, Chakrika?
Yes, sir.
Uh-huh.
So basically, air grafting is when we take a particular
Air
branch
layering,
and
layering. It's
oh,
layering.
okay, sir. Layering. Okay,
Mhm.
sir. So layering is a process where when we bend a stem, I mean a branch of a plant
Mhm.
near to the ground
Mhm.
and plant them, like we basically bend them into the ground.
No, you guys
So
haven't
like
got it. Look over here. Look here. Air layering now, guys, it is being referred to this over here. When we do it in the branches that are not close to the ground, branches um, in the canopy or in the upper portion of the plant. When we are twisting the part,
sir, I think
twisting
I got
branches,
it.
you got it now.
So it's like basically the same, but without the
Hmm.
involvement of the sand. I mean,
No,
like the soil.
there is involvement of soil. There is
So, but
involvement
like
of
it's
soil.
only the branch part which you're covering, right?
Look, there's a minor difference between the two. You are basically doing layering. Notice where I'm pointing. Here we are doing layering when the branches are made to grow in the ground. Yeah, the branches are made to grow in the ground. I mean, they are twisted close to the ground, right? And then they are covered with soil, and the adventitious roots grows here. But if you were to have a plant like this, say here you are having a plant and this is the branch, right? And now I am covering this part of it with soil, then covering it with polythene. After some times, what will happen? Roots will start to develop from it, and then I will be cutting it from here, and then I will be regrowing it. I will be replanting it. So that is called
Oh,
as air
okay, so I
layering,
got it.
right? So that is what air layering means. While I was doing it in this manner, where I was twisting the branch of a plant, okay, forcefully doing the layering, forcefully twisting it. So that was the case of the simple layering. Hope it is clear to all of you.
Okay, sir. Yes, sir, it's clear.
Okay,
Yes,
then.
it's clear now.
Right. What are some of the examples now? Think of some of the examples for which layering is done.
So jasmine,
Jasmine. Okay. What are other examples?
rose,
Jasmine is there. Yeah, for rose also.
So,
Interestingly.
mint.
Mhm. Mint. What do we locally call mint as? You guys
Uh,
give some more examples. Uh-huh.
Sir, I don't get it. Is mint not an example?
Mint is an example. What is the local name of the mint, by the way? Sundus, what is
Sir,
the
as in
local
like,
name for mint? Local name, like locally in different parts of India, like foods and foods have got different names locally.
Say you mean in Hindi?
In Urdu, in Hindi.
Pudina.
Exactly. That is uh, Pudina, right?
yeah, so
Mint.
same we say in Kerala.
It's same. That's the same in Kerala as well. Okay, okay,
sir,
Yeah, so
same in Telangana and Andhra as well.
okay.
wow.
That is Pudina. Okay, so these are some examples. Apart from that, you have example of black raspberries also. Raspberries. Okay, these are some of the examples in which layering is basically done. Okay, so remember those examples. What about those are the also examples like strawberry also. But then in the case of a strawberry, it we see natural layering. Okay, but for artificial, in the context of artificial vegetative propagation, these will be the examples of layering. Now we move on to the tissue culture. Tissue culture, I think the word should be familiar. Like tissue culture is what? Mhm. What is basically done here? Here you
Sir,
see.
well, the images, I think I've saw them, I've seen them somewhere, but I really don't, I'm not really familiar with the meaning.
Okay, okay, no issue. You might have study about the tissue test tube baby. Look here, tissue culture. So in this process now, we are
First
cultivating
time I got
plant
it, I
cells.
got it.
Got
Like
it?
the tissue, like they are grown in a laboratory.
Exactly, exactly. Tissue culture, tissues that are being grown in laboratory, right now. And we will be doing, we don't do tissue culture for every kind of plant, for only those plants that are endangered species of the plants that are at the that are in the endangered list. For those plants, tissue culture is done, right now. So look how it is done. Basically, in this process, we are cultivating plant cells. So you'll be taking plant cells from various regions of the parent plant. Okay, now. So you have just taken a tissue sample here from this parent plant. The tissue sample could be from leaf. Okay, it could be from root. It could be from a stem. It could be from bud. It could be from flower as well, right? So you have taken samples from different regions of the parent plant. Now you will be sterilizing it. You will be sterilizing it and uh, keep it in a specific medium So see here, tissue sample is placed in a in an agar growth medium containing nutrients and oxygens. See what happens now. I will be taking some sample of tissue. Now I will place it in a special medium, and this medium is rich in different types of nutrients. Okay, these are rich in different types of nutrients. Now after some times, now what will happen? It will develop into a mass of cells, right now. So that mass of cell will be called as callus. Okay, so callus, once you have taken some tissue from any part of the plant, you will be placing it in a special solution. Okay, that will be a special medium that will be rich in different types of nutrients. So thereby the cells will start to grow. After some time, the cells would, since it has started to grow, so there will be a mass of cells that will be called as callus. So you had this much of tissue, now it has grown to this much shape, right? It has grown like this. Getting it now? So this is now called as callus. So now callus, later on, it will be grown in a medium that consists of different types of hormones. What I said, I will be taking it in another medium that consists of growth hormones actually, right? So then you can see like this diagram, third one here, you see the tissue has now grown into tiny plantlets. Yeah. And later on, the plantlets are planted into the compost. Compost is basically a type of organic manure. Hmm. So here, what will later on what will happen when these mature into the full plants, or when they would have matured into the fully grown plants, then it could be transplanted into the main field. So for plants like the example of oil palm, for pine, for eggplant, okay, for like date plant,
Potato.
potato also, okay,
Sweet potatoes, is that right? Or just potato?
just potatoes, okay, potato, or you could be for sweet potato also it could be done for potato, for sweet potato,
So
right?
also banana, right?
Banana, yeah, banana is also there, and I think it's tomato is also there, right? So for these you get to see that tissue culture is done. Yeah. Is it clear to you guys or not?
Yes,
Mm-hmm.
sir.
Yes. So, Vaika, Chukrika.
So can you just explain it again?
Okay,
Yes,
look
sir.
here. No issue. See, first of all, the question is that what is a tissue culture, especially about the plant? Okay, because the manner in which tissue culture is done for plants and for animal cells, it is different. Okay, see
Okay, so
here, we are taking plant cells from different parts of a plant. Okay, like we can't be cutting cells now, so we basically say we are taking tissues, right? You can't
okay, so
literally cut a cell from anything. So then what I will do now, the process is such that I will be taking plant tissues, right? And then I will be putting it in a spatial medium that will be rich in nutrient, that is rich in nutrient, and I have placed it in the sterile environment. What is sterile environment? An environment that is made free from contamination. Okay,
okay, so
so since I will be growing a plant, I will make sure that there are no pests, there are no insects, right? There are no microbes. So I will ensure that these things are not there to harm the plant tissue, right?
yes, sir.
So I have placed plant tissue in a spatial medium that is having so much of nutrition in it. right? And now I have maintained an appropriate growing condition for this plant, right? Now this plant tissue, now it can be induced with certain types of hormones. I can introduce certain type of hormones in it in order to boost the development of the plant, right? I will be injecting this plant, I mean to say, I will be providing hormones that will increase the shoot growth, or that will also promote the growth of root system here, right? Now,
Yes, sir.
so now after some time it will grow into tiny plantlets. Oh, plantlets is clear to you. What is plantlets? Tiny plants are called as plantlets, right?
Yes, sir. That's
Okay. After some times, now I will shift those plantlets into a medium that consists of manure, right? So manures are also different types. Yeah. So I have placed it in compost. Compost are now basically manures prepared from the dead and decaying matter, and then you have got certain microorganisms in it, which are the good microorganisms that helps in the plant growth. Yeah, Chikrika, getting it now? Sandhu Spiker.
Yes, sir.
yes, sir. Got it.
So you basically took some plant tissue sample from any part of the plant. You have put in a medium that is rich in nutrition. Then you have provided suitable type of hormones to allow the growth of shoot and root. And then you will later on place those plantlets in the compost so that it starts taking nutrition on its own. Getting it now? So now then, once it would have grown to a required amount of size, then I will be planting it in the field or in the or a small land, right? Now, so this is what tissue culture is all about, right? Look, what are some of the is that clear to all of you or not?
Yes, sir.
Yeah.
It's clear.
Yeah,
Clear.
Chakrika, clear?
Yes.
Vaika?
Yes, sir. It's clear.
See, so basically the concept here was that the basic aim was to produce a very high number of plants, right? So look at, just you have taken a tiny sample and now we are able to get a very good amount of plant, right? So to produce a higher number of plants, this method is used. Okay. Second thing is that this method is used for especially for growing those plants that are endangered plants, right? Used for growing endangered plants. Just like animals could be endangered, plants could also be endangered, right? Why we'll be using this particular method for growth of endangered plants? Because I am providing a sterile environment that is safe from different types of contamination. So thereby, I will ensure that the plant grows in a contamination free environment, and as I will be having a higher number of plants, right? Third thing here we get to see the plants that are growing now. It will be similar to the same parent now. Here, the plants that are growing here, they are similar, and in similar in what manner? Genetically similar, right?
Yes, sir.
So both of basically the plants are clone of their parent now, right? Suppose if the parent plants were taller, so the clone plants will also be taller, right? Okay. So this is the basically the crux of the tissue culture. Anyone has any doubts, please do let me know.
No,
Also,
No,
like
sir.
you have to maintain a very adequate amount of temperature. We ensure that temperature is good here. We ensure that humidity is also good. We also ensure that it is getting proper amount of sunlight also. So this is all about the tissue culture. So we have basically covered the different methods of artificial vegetative propagation, that is cutting, layering, grafting, and tissue culture. So basically the first part of the chapter, that is the asexual reproduction in plants, we have covered that. All right now. So that thing we have covered in the chapter. Next thing
Okay.
we will be discussing about, it will be about the sexual reproduction in plants through flowers, right? So from here we will continue.
So, but before that, we have to complete like binary fission, right?
That's a small topic that will be covered under a minute or two. Okay.
Okay, sir.
Sir, I
Mhm.
know it.
Okay, sir.
Like it's very easy, that
Hi.
topic. I have
it's,
known it.
it's you have studied that. Very good.
Yes,
Okay. If you like, you can explain it to others.
like the process of the what do you call that? Ammonia splitting into daughter cells is called binary fusion.
Very good. Also, the daughter cells will be identical to the parent cells. Yeah.
Okay, sir.
Okay. Look here. Um, we have 1 minute remaining. Look here. You have a parent cell like this, right? This is the nucleus of the parent cell. This is the cytoplasm in the parent cell. Okay. At least you should be knowing this much, like a typical cell will be having nucleus and cytoplasm in it, right?
Yes, sir.
And the DNA, you all know about DNA. So DNA is basically found in the nucleus, right?
Yes, sir.
So plants, the microorganisms like amoeba, that is a bacteria. It will first, what will happen? The nucleus will start to divide like this. So DNA replication is taking place here. DNA is separating here and replicating into two. Second, the cell will become like this. Okay. Now what is happening? The cytoplasm, it is also separating. Okay now. DNA, that is the nucleus, is already separated. Nucleus has separated here. And then later on, the cytoplasm will also be separated. So you have got now two daughter cells. Two daughter cells, and both of them are identical to each other, right? And they were identical to their parent as well. Basically, the parent cell has actually split into two daughter cells, right? Uh, so I know it is not clear to you guys, but when I will be covering that in detail now, then we will be discussing each and every step in it. Okay. As
Okay,
for
sir.
now, just
Okay, sir.
understand that it is splitting into